Good afternoon, everyone. I am Jan Klapwijk, and I am the chair of the Toxicology Specialty Advisory Committee of the Royal College. I'd just like to thank you for joining us for today's webinar, which is called Establishing Mode of Action and Human Relevance of Human Carcinogenetic Outcomes. The webinar will be about 1 hour and will include a 30 or 40-minute presentation, and then we will have a short Q&A session. Please use the Q&A function at the bottom of the screen for your questions, and please feel free to start putting those in at any time. Attendees can self-credit 1 CPD point for attending today's webinar. The webinar is being recorded. So today, the speaker is John Foster, who is very well known to members of the toxicology pathology community, so I'll keep my introduction very brief. John's been working in the field of pathology since the late 70s. He obtained his diploma of the Royal College in 1987 and then became a fellow in 1997. And he's also been the chairman of this specialty advisory committee that I'm now chairing, as well as chair of the examiners for the toxicology specialty. He's also been president of the BSTP and is a fellow of the British Society of Toxicology. As well as this, he holds various visiting lectureships and honorary fellowships in different learned societies and universities. John's published over 150 research papers, review articles, and book chapters, and has been editor-in-chief of the journal ToxPath from 2008 to 2013. After working in agrochemicals and later pharmaceuticals, John became an independent consultant in 2013, where he is now working. So over to you, John. Thank you very much again for doing this talk.
Thank you very much, Jan. Welcome, everybody. When Jan asked me about the possibility of providing a webinar, I was about halfway through actually using this mode of action human relevance framework for a client. And I'm a great advocate for this sort of approach. It provides a number of advantages over what we used to do in the bad old days, where we would argue opinion in terms of whether or not, a carcinogenic outcome was relevant to humans. But this approach, I've done it a few times. The first one I did was about 5 years ago when I was working with Mac Proven in Regulatory Science Associates. Since then, I've done about 5. This last one that I recently finished was the latest. So what I wanted to do was to try and the approach can be a little confusing. If you look at the guidance that's available, there is some good guidance. There are some good templates for when you're preparing this sort of approach. But I thought it would be nice to give you an introduction using one case example. I did say I would use more than one, but there isn't really the time for this. But I'll go through the process itself, and you can see if you like it, I would love that. If you don't like it, then I think you're missing a trick, but there we go. Maybe we can decide that by the end of the presentation. So the next slide then is a history of the concept. It was voiced or proposed in the late 1990s to early 2000s, originally in America by the Environmental Protection Agency and the International IPCS. And then throughout the 2000s, it was modified through a number of workshops, and a lot of academic people were involved in the discussions, some regulatory people from agencies throughout the world, including Europe and the USA, and also people from industry. Primarily, the target for this was agrochemicals, so pesticides and herbicides, But since then, I think it has shown that its value can also be extended in terms of looking at drug-induced effects as well. There's what I've put on this slide here. There are two websites that I think you should look at. WHO is the auspices that IPCS works under, and you should look at these two websites to see the processes. And there are good links to other sites, including ECHA, the European Chemical Regulatory Institute agency. So let's ask the question, why might we need this approach? Well, this is an EPA database. This is from a presentation that Vicky Dell'Arco gave in 2008. So this is from how many? 219 pesticides, and these are the carcinogenic outcomes. You can see that for the mouse, the liver is the major organ, and followed by the lung. For the rat, it's thyroid, testes, and liver. So the question is, in every single one of these 2-year bioassays, how do we decide whether this is test article related and what's the relevance of this effect to humans? Because originally we accepted that all animal outcomes were relevant to man. Then we went through a situation in the 70s and 80s where we tried to disprove that any of this was relevant to man. Much of that was based upon opinion, but the regulatory outcome for most of this was, in the absence of information to the contrary, the default is to assume that every test article induced neoplasia, and you do have to decide whether it's test article related, of course, but every test article induced neoplasia will be relevant to human tissue or human exposure. Now, clearly, this is a naive approach, and it would result in a lot of useful chemicals not progressing. So why do we need mode of action human relevance framework? Why do we need this? It provides transparency because it takes you through a statistical and scientific weight of evidence evaluation for the basis of your decision making. So it's not the loudest voice anymore. It's a scientific approach. It ensures rigor in the evaluation and consistency of your documentation. Where do you get this from? Is it from studies itself, or is it from the literature? It aids, and you will see this as we go through the talk, it helps in identifying where we have critical data needs or research needs. So you will not generally be able to develop one of these mode of action HRF framework. You will not be able to get it from a single study. It will require a number of studies, and in some ways a considerable amount of extra work. But it does provide a basis for a dialogue between risk assessors and researchers. The problem in the past has been that while researchers have come up with some very interesting data, getting the risk assessors involved at the right stage meant that much of this work was ignored, and the default, especially in Europe, the default was to go back to this being any effects in animals being relevant to man. Part of the process requires a sufficiency of evidence, and it's a jargon, of course, and if you look through, that might not immediately occur to you what exactly that means. But it requires expert judgment, peer engagement, so you have to try your mode of action, especially outcome. You have to make sure that it's robust, and thorough review. You will see that as we go through, but there is increased regulatory acceptance of mode of action human relevance because it is seen as a structured, scientifically based rationale for any decisions or any debate that goes on in terms of the decision. Now, there was initially some confusion between what's the mode of action versus the adverse outcome pathway. And many of you will be familiar with AOPs. Generally, mode of action is chemical specific, whereas AOPs are generally generic. So you don't develop an AOP for a single chemical. It tends to be developed for specific classes, but there are exceptions. PPARs, thyroid hormone dysfunction, etc. These are things that AOPs have been very well developed for. But there was some confusion about the two approaches with a concept that they were exclusive. So you were either in favor of preparing a mode of action or preparing an AOP. This is wrong. They're both parts of the process, and they are certainly both integral parts of developing the mode of action human relevance framework. The AOP, through similarities, will generally construct the initial part of the mode of action, which will subsequently involve an evaluation of the weight of evidence for the strength and plausibility of the hypothesis. I can come up with all sorts of hypotheses, many of which will be harebrained. But if you use the mode of action HRF pathway, then you will see that you need a little bit extra evidence, or you need to generate that extra bit of evidence in order to increase the strength or plausibility of your hypothesis. And you'll see this from the case example that I will show you. So AOPs and mode of action, they are complementary, and you will need to use both seamlessly. And you will see that much of what I will show you for the mode of action follows exactly what you'd expect to see if you were looking at an AOP. Okay, how do you generate your mode of action? Well, the most obvious one is through a GLP study. And when we're talking about carcinogenesis, it will be generally a two-year study. So the GLP two-year study will be there. Pathology will be important, but as you will see, it's not going to be sufficient on its own. Pharmacology and knowledge of the pharmacology of your compound, not always there for agrochemicals, of course. A knowledge of how similar chemicals have been discussed and used in terms of working out their mode of action. Target distribution studies, literature data, it's always important to support your hypothesis by published data. Non-GLP studies, there's nothing wrong with these, and GLP studies for carcinogenesis that have been conducted over 14-day, 28-day, 90-day studies. So all of these will be integrated, these approaches, to come up with your hypothetical mode of action. So we have kidney cancer. This is a postulated mode of action. This isn't one of the case examples. This is a generic approach that you would have. So we get tubular carcinomas in the kidney. We have both 90-day studies and 2-year bioassays that show that the kidney is a target. We have found metabolites, for example, in the urine, mercapturates, so that's telling you that glutathione metabolism is involved. We have decreases in glutathione, for example, in the liver, and radiolabeled studies here. We can see that it's bound, radiolabel is bound within the chemical for a radiolabeled compound. So you're not going to get many of these things from a 2-year study alone. So we believe the hypothesis we came up from this data is that the chemical was metabolized in the liver and bound to glutathione. It enters the blood, is filtered at the kidney, concentrated and absorbed within the kidney. We see degeneration. Here is a section of kidney with some S3 tubular necrosis. That's accompanied, degeneration accompanied by regeneration, diffuse hyperplasia, focal hyperplasia, atypical hyperplasia, tubular adenoma, and carcinoma. So this is our hypothesis. From degeneration onwards for the kidney, this can be applied to all sorts of different organ systems where carcinogenesis is the outcome. So this is not specific to the kidney. The earlier parts are. Okay. So what the mode of action HRF approach asks you is 3 questions. The first question is, there's sufficient weight of evidence to establish the mode of action in animals. Question 2, are there key events in the animal mode of action that could work in humans? So is there qualitative similarities between these? And then the last one, can human relevance be excluded on the basis of qualitative and quantitative differences? Because sometimes the amount of a particular chemical might be too low to generate sufficient of the metabolite needed in one of the key events. So the steps that you need to take when you're considering this, you formulate the problem. This is not a trivial exercise. And for some problems, it will not be possible to actually adopt this. It would be too much work. Hypothesize mode of action. You need to develop that. Summary of the data used. So you have to explain which data you're using to establish which key event. Listing of the key events identified. I showed you that there. Bradford Hill considerations. You'll hear me refer to Bradford Hill. He was an individual, an epidemiologist. He produced a paper in 1965, which when they were discussing this approach, they used a lot of his considerations in terms of dose-response relationships, temporal association, consistency and specificity, qualitative and quantitative human concordance. These things don't mean an awful lot with me just describing it to you, but we'll go through a little bit more detail. Consideration of alternative modes of action. There are, in terms of a kidney carcinogenesis, there are a number of possible modes of action. What uncertainties or inconsistencies are there in the mode of action? Any identification of data gaps, and then conclusion in terms of the human relevance. This is in ECHA, on this ECHA website here, and I recommend you read that. So the postulated mode of action, the ticks are where we found some data. There are some bits of data that are missing from these, but you can see that the molecular initiating event, we've got data for that. The key event, the single key event, we have data for that, and for subsequent key events. So we're in a pretty good position in terms of a mode of action for producing cancer in the rodent. This is the concordance. What you're looking for is dose responses. So we need to see the key events increase in intensity or severity as we go up the doses. At time, you will see that at low dose levels, only some of the key events will be present, whereas the later ones won't. So for a non-genotoxic carcinogen, this is what you'd expect. You'd expect some thresholds here. So at low doses, you see the early events, but you don't see the late events. And then at the carcinogenic dose level, you will see all the events. Some of them will occur very early, and some of them will require the 2 years. So strength of association, for each of your key events or for your molecular initiating event, you will assign a confidence, and that's from high to moderate to weak. High confidence is when you have experimental data that shows a particular event occurs, and low confidence is when you may not have the event there, and that clearly will identify areas where you may have to generate extra data before you're ready to submit this to a regulatory agency. So what about consideration of alternative modes of action? This is Sherlock Holmes, of course. So when you've eliminated the impossible, so that's the alternatives, whatever remains must be the truth. Not quite that black and white for this approach, but it goes along with that. So if you look in terms of alternative modes of action for inducing kidney cancer, there's mutagenicity, of course. And this would be there for all of the, for carcinogenesis in any of the organs that you see in animals. So you need to be sure that you've got a good, robust GLP package for mutagenicity, and then the answer is either yes or no. These are the others: chemical exacerbation, alpha-2 globulin nephropathy, for example, and cytotoxicity. Each of the modes of action, if you look at these, they have a set, an association. So some of them are gender specific. So alpha-2 globulin is only seen in male rats. So if your results show mouse and rat, then clearly it's not that. You'd need to generate a similar listing if you were looking for other organ systems. So this wouldn't be the same listing for liver, for example. And as I say, genotoxicity needs to be generic. So let's go to the case example that we've got. This is chlorothalonil. It's a fungicide. When ICI bought Stauffer Chemical Company in the late 1980s, I worked a lot on this compound. It was a very important compound. It's still marketed even today. The toxicology and mechanism of action have been extensively studied, and during the past 20 years, a number of reviews have appeared by scientists and expert panels, including the mode of action HRF approach for this. This is what you see early on in the first week or so. So you've got hyaline casts here, and you've got single cell apoptosis or necrosis within the kidney. Nothing in the liver, nothing in any other organs. But if you look, because this is an old compound, there's been a number of different carcinogenicity studies, certainly in the late 70s and throughout the 1980s, in various rat strains, Osborne, Mendel, and Fisher, and CD1 mouse as well, some 24 months, some 18 months. So if you look in terms of neoplastic events in the kidney, in the Osborne Mendel, these are the doses. So at the controls, 0 out of 62, and then 3, and then 4 in the males, and in the females, 0, 1, and 5. So not a great response, but still a convincing response. If you looked at higher dose levels for the full 2 years here, then you've got 0, 7, 7, 19 in the males, and then 0, 3, 6, 23 in the females. That's pretty convincing, and similar effects you saw in the 30-month study that was conducted in 1989, but at lower dose levels, because these dose levels were there to look for no effect levels. In the mouse, similarly, we got lower response, but still a fairly convincing response in males at these dose levels, much higher dose levels, of course. At the lower dose levels, we didn't get, in the CD1 mouse, we didn't get kidney effects, but we got forestomach effects. But chlorothalonil, the conclusion was it was a rodent kidney carcinogen. But this has been widely used for some years, and as I say, is still used now. So we wanted to see what the human relevance might be. So the mode of action for renal carcinogenicity, we got glutathione conjugation. We could see this because we measured metabolites, so some biochemistry involved in this, some mercaptorate metabolites in the urine. We got accumulation in the kidney, and we could inhibit this accumulation by treating animals with this inhibitor of the anionic transporter system, probenecid. So we could stop that, and if we used probenecid, we didn't get kidney necrosis. We had evidence from analogous compounds, not from chlorothalonil, that CS lyase or beta lyase was the activating compound, but it was a bit of data that we didn't have. We got renal toxicity, consistent time and dose histopathology, and clinical chemistry showing renal toxicity. We got proliferation within 7 days with bromodeoxyuridine, and we also got tubular hyperplasia that we could see in the longer term studies. And then we got adenoma and carcinoma and increased incidences in these situations. So was the weight of evidence sufficient? Well, we go back and we do this concordance, 24 hours up to 2 years. So at the low dose, No, we didn't see anything. At the higher dose, we saw the early key events up to metabolism of thiols, but we didn't see anything else. And then at the carcinogenic dose levels, 40, 80, and 175, we saw all of the key events, including the neoplasia. So alternative modes of action, it's not genotoxic. We had done a very robust panel. We got generation of GSH conjugates, so that's yes. CPN exacerbation not present. Alpha-2 globulin not present. Indirect DNA reactivity, we saw no evidence for this, but we did get cytotoxicity. So we actually got 2 of our modes of action that were interacting to produce a cancer. So human relevance, GSH metabolism, evidence in animals, yes, and probably we thought it would be relevant in humans. Cysteine conjugate uptake, yes, probably. Metabolism to cysteine conjugates, probably in humans again. Renal toxicity, we thought probable. We didn't have evidence, but we knew the enzymes were expressed there. So chlorothalonil causes renal tumors in the rat via formation of a cytotoxic thiol metabolite, resulting in sustained regenerative hyperplasia. So we had a pretty high level of confidence. This is a paper by Chris Wilkinson and Jim Killeen in 1996. They did an awful lot of this work as consultants and from the CRO. So risk of renal carcinogenicity in humans, any qualitative differences? No. But there were quantitative differences in the expression of some of the key enzymes. Yes, so renal carcinogenicity in humans, yes, but the mode of action has a threshold for a number of the key events, and humans were considered to be far less sensitive than rats in demonstrating the key events. So I said there wasn't an awful lot of direct human evidence, but there is a case study, and this appeared in 2017 here. It was a 43-year-old man who ingested daconil. The fungicide contains 30% chlorothalonil, and about 12 hours after ingestion, the patient had acute renal necrosis, although the function recovered, and acute toxicity, this is a conclusion from this case report, described very rarely. No previous cases have been described. So this is a decision tree for using the mode of action. Is the weight of evidence sufficient to establish a mode of action in animals? If it isn't, then the default is to assume that it's relevant to humans and continue with the risk assessment in the USA or classification and labeling in Europe. If the evidence is sufficient, then are there qualitative differences in the key events between animals and humans? If yes, if there are differences in enzymes, for example, then you assume the mode of action is not relevant and there is no risk assessment and no classification and labeling because it's not relevant. But if there aren't any fundamental qualitative differences, you then go on to the last question. Are there fundamental quantitative differences in any of the key events? Yes, there are quantitative differences, then again, you assume the mode of action is not relevant and you get away no classification or labeling. If no, then then you assume that the mode of action is relevant and you continue with risk assessment and classification. So what about regulatory acceptance? This is the ECHA website. It mentions the use of mode of action human relevance here. So they are they're acquainted with this. So WHO framework is referenced in EU guidance, in REACH, in classification and labeling of biocides. It's applied in Canada and USA, and it's in their regulations. The concept is used to affect labeling. So you can change labeling if you have not relevant, if you show not relevance to human, then there's no labeling, of course. But you can downregulate if you can argue that conditions are fairly specific to the rodent. So again, I would recommend you look at the ECHA template. You must assign yourself to the template because that's what ECHA will expect. That's what the Committee for Risk Assessment, the RAC committee, will expect to see. This was from a presentation by Maria Pronk. It mentions site of action and mechanism or mode of action studies. When you have such information, in a lower classification may be warranted, or if there's scientific evidence that supports that the mode of action is not relevant, then the substance won't be classified. And again, if you look at the ECHA site, that you will find this. What about examples of changes in opinion in Europe? There are some for pyrethroids. For this one, metafluthrin, this is liver tumors. CAR activation was considered or argued to be the most plausible mechanism. Now, the ECHA Committee for Risk Assessment considered that this mode of action could be relevant to humans. I know many people consider that it's not, but some people do consider that it is relevant. However, the critical key event in the mode of action for liver tumors in rodents was the induction of proliferation. This was not observed in human hepatocytes in vitro. They were treated with the metabolite. They didn't show proliferation, and the RAC concluded that a classification was not justified. So it went from a potentially classification type 1 category through to no classification at all. So RAC considered. They subsequently evaluated several other compounds for which the mode of action involves the same pathway, and concluded that it was not relevant to humans, again based on the lack of proliferation in human hepatocytes. And there was no carcinogenicity classification for 6 of these 10 compounds, and a down classification from category 1 to category 2. So this approach does have regulatory acceptance, and it does have impact if you do it properly and appropriately. Yeah, I'm going to leave that one. There are some literature references at the end of the talk. I'm not going to go through them all, but please, I recommend you look at these. They give you examples of how to apply the approach, and I say you can go through the guidelines in terms of templates, etc., but it's nice to see some positive examples. I'm going to stop there. Thank you for your attention. I'm sorry I've gone on a little bit longer than I intended, but please ask questions, and I'll try and address them. Thank you very much.
Thank you very much, John. No, you haven't gone over time at all. We've got plenty of time for questions. So just like to invite questions from the audience. As I say, please use the Q&A box at the bottom of the screen. Just a quick question from me, John. I mean, if I understand correctly, this isn't, if you like, a regulatory requirement, but are most people doing this now, or is it really...
It depends how important your compound is.
Yes.
But where you get... It's there to address, although I've talked about carcinogenicity, you can also use it to address non-cancer endpoints, non-neoplastic endpoint, toxicity, yes, because it goes through the same argument in terms of relevance to man. So if you've got an important chemical that you need to defend, that you need to evaluate in terms of its relevance to human exposures, then this approach is being used. The large large agrochemical companies, Syngenta, Bayer, etc., they are using this approach, and they've been very successful. Not in all cases, because in some cases there are data gaps that can't be addressed, that can't be fulfilled. But it is important in in generating or stimulating research into these things. And a knowledge of other people's compounds, and publishing is important. I know some companies are reluctant to publish, But if you don't publish, you will not be able to use data that's already out there.
Right. Okay, thank you. We've had a couple of questions come in. I think the first one from Chris Elcombe, I think you've just answered. Chris, if you disagree with that, please ask again. But you were asking about non-carcinogenic adverse outcomes. And then Richard Howarth asks, he thanks you, John, for the presentation and says, what's the most frequent data gap in your experience in developing these MOAs, and how have you advised clients in addressing those?
Yeah. The most frequent gap, many of the key events that you saw that I showed you are common. They are generic. In terms of carcinogenicity, cell proliferation is key.
Yeah.
So you will have to generate that. You won't get that from a 2-year study. You can, of course, you can argue by inference that basophilic cell hyperplasia, for example, reflects cell proliferation, but you may be turned away for that. But the most common area where there are gaps is probably the molecular initiating event, the first event.
Yeah.
Some of that, it can be difficult to generate. Some laboratories are there to establish it. Some of them are CROs, and the CROs are offering more and more of this sort of investigative approach now. It used to be the large companies like Syngenta, like Zeneca, like before them ICI, had their own laboratories that could generate this. Generally speaking, that's not the case anymore. But there are laboratories out. I'm glad to hear that Chris Elcombe is on the call, because his father, of course, Cliff Elcombe, was instrumental in getting particularly the human relevance aspect, the in vitro aspect for this. And in terms of the pyrethroids, for example, the pyrethroids would never, the mode of action was fine. Establishing CAR relevance was fine. But unfortunately, because of the conservative nature of regulatory agencies, there was enough doubt out there to question whether CAR was really a rodent-specific phenomenon. But the lack of response to human hepatocytes for cell proliferation to these chemicals convinced them that really humans were certainly quantitatively much less responsive than rodent hepatocytes, for example, for that particular event.
I mean, are regulators generally reluctant to use a quantitative approach? Are they much happy to see
well, it depends whether you're in Europe or the USA. The USA have always been very, very good with quantitative approaches, whereas for Europe, of course, qualitative hazard evaluation was their most important concern. But it is changing. And on the RAC committee, there are scientists. They're all scientists. They're not full-time regulatory individuals. So, I mean, I think that the buy-in, and the reason they bought in was that they were involved in developing the mode of action human relevance framework in Europe. But I've done one drug, I've done one pharmaceutical using this approach. And it really provides you with solid evidence that you can go and discuss these things with pharmaceutical drug agencies as well. And it gives a very clear transparency onto why you are saying this. You're not saying it because, oh, mice are mice, aren't they? And they're extra reactive. You're saying it because you've got the evidence there to prove it.
That makes a lot of sense. But a couple more questions coming in, John. So, anonymous question: will a case report be accepted as evidence, bearing in mind that they wouldn't be as good as a controlled study with large numbers of people or animals?
No.
You're shaking your head.
No. It has to be scientifically robust, and I'm sure the person who asked this knows this. A single individual, you very rarely know, for example, what dose level they show. The time involved is variable. Treatment, it concentrates the person. It changes the way a person can react. So no, case reports are not robust in this way.
So an interesting question from Leslie Stanley, which has been backed up by Sue Hubbard. Great presentation, John. Leslie was wondering how this approach can inform the thinking about this balance between hazard and risk-based assessments, especially bearing in mind the different attitudes between these two in different regulatory jurisdictions, which you've obviously touched upon. But do you think this approach will somehow help change the approach in one or the other?
I mean, it has helped. It's removed classifications in Europe, but you do have to have, I mean, for those chemicals, there's been for those pyrethroids where it has removed the classification category. There's been a huge amount of work, and it has actually probably taken, I don't like to say this because it's going to turn everybody off, but it's probably taken 30 years of generating data for these compounds. Pyrethroids are very old pesticides, of course. The ones that I've been involved in recently over the last 5 years, clearly that hasn't taken 30 years, but equally I couldn't hold my hand up and say that these have worked.
No.
But they provided me with lots of money.
Yeah, very important. Anna Lanzoni asks, how does human inter-individual variability get, I guess, incorporated into the MOA framework? How do you consider that?
That's a really good point. And if you have indications that a particular enzyme that might be key to your, mode of action, if that particular enzyme is expressed in a rodent fashion by a subset of humans, then that would be taken into account when changing the classification. But I still think you would likely get if you prove that a particular enzyme was absolutely key to generating a toxic metabolite, then I still think you would likely, if that was very rare in the human population, you would likely get a down regulation from a category 1 to a category 2. You probably wouldn't remove, the category classification category totally, but it would be important to know, and you would probably be asked that question. I've never been asked in any of the ones I've been involved in, but
important, isn't it?
Yes.
Another interesting question: can we use, from Yogananda Mulamath, can we use other species, alternative to rodents, to assess carcinogenetic potential in small lab animals? If so, which species? And for instance, some metabolites are generated in the hamster. And supplementary question about duration of those studies, but really it's about other species. I mean,
where people have done this in the past, they have used other species. The hamster has been one of the species that they have done, but they don't, generally speaking, I wouldn't advocate you doing a carcinogenicity study in another species. But what you might use those other species as, they would be sentinels for you to test your hypothesis. Because if you believed that a particular PPAR was involved, for example, and the hamster didn't show it, then you could use the hamster as one of your blocking. And actually, the guidance suggests that you try and intercept your key events to see whether or not to assess the robustness of your key event, to block that key event. We did this with probenecid. In terms of that, we blocked the next downstream key event, which was accumulation, and we didn't get toxicity. So I think if you were going to use other species, then yes, I think you could use them in that way, but not to do carcinogenicity studies on. And the real reason for it, we have difficulty interpreting the data from rat and mouse carcinogenicity studies as it is. Now we've got the transgenic, we've got the 6-month transgenic mice and the 9-month transgenic mice, and even those, I have been called in on occasions to help interpret data from that, because they are not as clear cut as perhaps they when they were originally proposed. They are getting tumors, they are getting spontaneous tumors during the 6-month and the 9-month durations. So no, don't do carcinogenicity studies in the rabbit or the hamster, but certainly interrogate your mode of action by using other species to test a particular key event.
Okay. Another interesting question from an anonymous attendee is about any thoughts about how AI can help to do these practices more rapidly. You've still got to generate the data, haven't you? But how artificial intelligence might be able to help. Any thoughts?
For me, the artificial intelligence is equivalent to you doing proper literature searching, because that's where you're going to that's where you're going to use. You will ask artificial intelligence, please interrogate the databases for these sorts of key events. So yes, I think they will do that. They probably do it more efficiently than you or I could do searching through the literature. And they could also help you come up with modes of action by picking out key events that are relevant to the particular types of chemical. But, in order to get your mode of action, you do need to know quite a lot about the chemistry of your compound. And for some compounds, we don't know an awful lot about them. It's very different if you look at drugs versus agrochemicals. With drugs, you know the target, you know the molecular target, and clearly not all toxicity or endpoints like carcinogenicity are related to the target. They could be off-target effects, but still, I think you can eliminate some of the things by a knowledge of the pharmacology of your compound. But if you're developing a small company, developing a single product, that can be quite difficult to find. But there are structure-activity relationships that you can look at similar chemicals to see how they work and how they interact with biological systems.
Yeah.
But I don't think we should exclude any of this sort of.
No, absolutely not. So I've got another question coming in from George Cass. Great presentation, John. Do you think the inclusion of endocrine disruption as a new class in Europe's CLP will impact the use of quantitative information to characterize the relevance and the risk to humans?
The answer is yes. I mean, I think it will do, because, again, not that I've ever seen any examples. Maybe George has got some examples, actually, of where the approach has looked at endocrine disruption. But there's no reason why it shouldn't look at endocrine disruption, and particularly where I have been involved in looking at an endocrine-dependent organ, the mammary gland, for this sort of endpoint, and developing a mode of action for that involves disruption of the endocrine system, including the pituitary and ovary, etc. So yeah, I see the approach. I see no problem with using this sort of approach to work out that sort of thing. And of course, they assume that an endocrine or an effect in an endocrine organ is endocrine disruption. That's not always the case.
No.
There are other ways in which you can get changes in endocrine-dependent organs that are not endocrine-dependent. The change is not endocrine-dependent.
Okay, sorry, a couple of questions coming in, and we've got a bit of time. But Anna Lanzoni, you probably know, asks about.
I do know Anna.
Yes, exactly. Well, I think it's not really a question. It's more of a comment about perhaps better investing in vitro and NAMs. To get it, we'll lead to better regulatory acceptance rather than just testing in vivo. And it's not really a question, I think, but
I think it's great, but I don't think you should limit yourself to in vitro or in vivo. You have to use the technologies that you need to use to support your argument. I think in many ways that's always been the case. It's not. If you're talking about NAMs for carcinogenesis or in vitro systems as surrogates for carcinogenesis, then you're talking to the wrong person. I just think that can't easily be done. But I'm sure Anna a lot of experience in the NAMs, whereas I can't really say that I have. But thanks for the question. It's a good one.
That's important. And then there's another one here. I'm just reading it. So once the MIE and the KEs are identified in animal models, how many of those KEs should be reproduced or not in human in vitro models to inform on the human relevance? So, and how are the limitations and validation of the in vitro models considered for this assessment? So it's really about how many of those key events do you need to reproduce to be able to determine whether it's human relevant or not using human tissues.
It depends what endpoint you're looking at. But if it's cancer, the key endpoint is cell proliferation. You cannot get cancer without cell proliferation. So for that, the key event that you need to display or not display in humans is cell proliferation. So for cancer, that's it. For a cytotoxic endpoint, for a cytotoxicity endpoint, then it's probably generation of the metabolite that you need to demonstrate doesn't occur in. So it's not, you do not have to demonstrate every key event in order to prove your mode of action. Because if one key event is missing, it won't work. It doesn't matter where, if the molecular initiating event doesn't occur, the one that you've proven to be key, the molecular initiating event, if that's essential for everything downstream, if you don't show that in humans, then you can forget about it. That is not relevant. That mode of action is not going to be relevant. So no, you don't have to demonstrate every single key event is not relevant. You just need if you like, the very key event. Yeah,
so the better you understand the pathway in animals, the more easier it will to do that, to find that, common thing. Richard Howarth asked another question. It seems like metabolites are often very important in establishing an MOA.
Yeah.
What kind of evidence do you need to establish which metabolites occur in rodents and which are found in humans? Presumably from in vitro hepatocyte culture.
We did it. We did it by generating, so chemically manufacturing the metabolite, and then dosing either animals in vivo or cells in vitro, and showing that clearly it's possible that there is more than one metabolite that's cytotoxic, or that the metabolite that's cytotoxic is has such a short half-life that it's very difficult to demonstrate that. And that will be the case. For some compounds, you will not be able to demonstrate. So in those cases, you have to argue that, and this will be an area where you need to identify a shortfall. So there are very few modes of action where all of the key events have actually been shown. I showed you a real idealistic example of this. But for most chemicals, some of the key events won't be shown. But you will have to go through the literature to see what similar chemicals and how similar chemicals are activated and how they're cytotoxic and by what means they are, and you would test for that. So you might block a particular enzyme using an enzyme inhibitor, and you don't get toxicity.
Yeah, I mean, related to this, we've got a sort of an unknown type of question. So it says, is it not possible that once an MOA is proven not to be relevant to humans, the same adverse outcome may be realized via a different MOA?
Well, but no, but that's covered in this approach, because you have to consider what the other MOAs. But you're right, there may be an MOA we don't know anything about.
Yes, that's right.
For cancer, I think that's highly unlikely. We do know what the key events for carcinogenesis are. Cell proliferation is key. And that's generally accepted to be the case. So you may be producing cytotoxicity via a different mode of action to the one that you're proposing, but the response to cytotoxicity is cell proliferation.
Yeah.
Cytotoxicity is a potentially, it's a loaded one. It's a bit of a grenade in your hand, because you can produce it by potentially a number of ways other than perhaps the mode of action you propose. But that gives you that when you go through the way of generating this, it will give you some confidence. And if it's weak, if the association for that key event is weak, and that may be the generation of an active metabolite, then that will be shown. And when the agencies look at your response, they may be less able to accept that this is not relevant to man.
Okay. So we've got time for one more, I think. Chris Elkham's asked another question. So when investigating relevance by this framework, what method would you suggest? Bottom up, I start by confirming initiating event and work towards the adverse effect, or top down, or the other way around?
I don't think we've got time to address that. Sorry, Chris. But if you look at the references that I have, and particularly what we have is in the template, the ECHA reference template, there are guidances in there to assigning the strength of association between the key event and the subsequent events. So if you have a look on those websites, but I like the ECHA website. So I think if you do that, it'll answer the question. But it's a good question to ask.
Yeah, no, exactly. Well, look, we're more or less exactly on time, and I really appreciate all the time you've taken, John, to obviously give the presentation, and the Q&A I thought was excellent. So so thank you very much, and thank you to all the attendees for joining us. As I said before, the recording will be made available on the Royal College website, but I'm sure John will be open to receiving any questions offline if necessary. So with that, I will draw the meeting to a close, and thank you again for all the team at the Royal College for setting up the meeting, Carla and Kristen, and look forward to the next one. Thanks very much.